Okay students, we will begin with the structure of kidney. We have already seen that the kidney is located in the lower abdomen region, and it is located between thoracic vertebrae number 12 and lumbar vertebrae number 3. In humans, the right kidney is little lower as compared to your left kidney, and that is because of position of liver. Liver occupies the most of the space in right side, and that is the reason it is located little lower. A typical kidney is bean shaped. As you can see, the outer surface, this outer surface is convex and the internal surface is concave. The kidney is covered with renal capsule. The kidney is covered with renal capsule. Here it is shown in white double layered structure. Inner to the renal capsule, that is this portion, this gray colored portion, it is referred as renal cortex. Renal cortex, that means kidney is divided into two regions, that is renal cortex, and inner to the cortex you can see these green color structures which are similar to a pyramid or triangular structure. These are referred as renal medulla. This region is referred as renal medulla, which includes medullary pyramids. Now as you can see, there are strokes shown in the pyramid region. These strokes are actually created because of loop of Henley, which I will be discussing soon. Loop of Henley and collecting duct. So the medullary rays in the medullary pyramid is because of loop of Henley and collecting duct. So in this region the urine gets directed or collected, and then it is dumped into this orange color structures, if you can see, which is arising from the tip region of apex region of your medullary pyramid. These small minute tubes are referred as minor calyx. These are referred as minor calyx. So there are like 6 to 8 minor calyxes present, which fuse to form major calyx. As you can see, the darker portion of yellow color tube, this portion, this is referred as major calyx. So all the minor calyx will collect the urine, and they will dump or they will lead this urine into the major calyx. 4 to 5 major calyxes might be present in your kidney, which will fuse to form a single triangular, more or less triangular structure called as renal pelvis. As you can see over here, it is referred as renal pelvis. Now the tip or the apex region of the medullary pyramid is referred as renal papillae. Renal pelvis is the final portion or final part which will be receiving the urine. Once the urine is collected in the renal pelvis, it is sent out via ureter. It's a narrow tube and it sends the urine by peristalsis. I hope the internal structure is clear, and it's pretty much easy. You have to just remember about the two regions, that is renal cortex, renal medulla. Medullary region contains lots of tubes, and in sequence the urine travels from collecting duct. Collecting duct leads to the minor calyx. Obviously there is certain structures present between this. I will be discussing in upcoming portion. So minor calyx will lead to the major calyx, and finally the major calyx will lead to the renal pelvis. Okay, just a quick revision. It is divided into two zones, that is outer renal cortex, which contains malpighian body, PCT, and DCT. The inner medulla contains medullary pyramids, contains loop of Henley and collecting ducts. The urine formed in the kidney by the functional unit called as nephron, which I will be now discussing with you, and it is collected and released into the minor calyx. The minor calyx arises from the apex of the medullary pyramid. As you can see, I have drawn here a separate medullary pyramid which leads to the minor calyx. The minor calyx leads to the major calyx, which then fuses. Okay, just give me 1 second. Which then fuses to form renal pelvis. And renal pelvis finally exits as ureter from the kidney bladder. Ureter from kidney and brings urine to the bladder. Hope you understood the structure. Let us begin with the structure of nephron. But before starting that, we will first observe how ureters are made. I mean, how ureters are formed in the body and what are the different layers, and how urinary bladder exists. Let us start. Consider this is the transverse section of your ureter, which contains the outer layer. This one is outer layer. This outer layer is referred as tunica adventitia, or just adventitia. Then the middle layer is comprised of smooth muscles, which is muscular. Now this muscular tissue is again comprised of longitudinal muscles. Then you have circular muscles in between, and then again it is longitudinal muscles. Now this was the second layer, that is muscular layer. The innermost layer is similar to your digestive tract, that is the inner layer of the digestive tract, and this third layer is referred as mucosal layer. Mucosal layer, that is mucosa. So these are the three layers which is present in your ureter. And it brings the urine from kidney to urinary bladder. Obviously the mucosa surrounds the lumen. This is the lumen. Let us move to the next part, that is urinary bladder. So again, urinary bladder is a bag-like structure. And again it is made up of thick layer. If you have a close look at the urinary bladder, it has a rounded structure which is referred as fundus. So this region is referred as fundus. And internally, if you see, there is two openings. These are openings of ureter. Openings of ureters. So ureter opens here and it opens obliquely. Okay, oblique opening. So it collects the urine, and just hold on for 1 second. Okay, so the urine gets collected over this region. There will be sphincters present inside. So there are two types of sphincter present. There is one is internal one. So I'm drawing in blue color. Okay, this one is referred as internal urethral sphincter. Sphincters are rounded muscles which regulates the exit of urine. So internal sphincter is involuntary just give me 1 second. Involuntary muscle, so that means the regulation is involuntary. We don't have any control on this particular sphincter, but there is one more sphincter which is present on the outer side. This one is called as external urethral sphincter. This one is voluntary. So for the remembering point of view, you can just remember internal, IN internal and IN involuntary. So this will be easier to memorize. Just remember both muscles are different. One is voluntary, one is involuntary. Voluntary gives you the control to release the urine on your well. Okay, let us observe how the bladder is containing 3 layers in this particular wall. Yeah, how the wall is made up of in case of bladder. So we are going to zoom this portion, the wall portion. So the wall is made up of again 3 layers. At this portion, suppose it is urine, this side, that means this is the internal side. So this is basically made up of the inner side of the bladder wall is made up of transitional epithelium. So this, let us consider this is as transitional epithelium. This one is transitional epithelium. And you know that transitional epithelium is also known as urothelium, so that it can stretch, so it helps in holding the urine. But the main function of release of urine or to retain the urine inside the bladder is because of the second layer, and that is, just give me 1 second. The middle layer, and this middle layer is made up of smooth muscles. Okay, this is made up of smooth muscles. The smooth muscles are specifically, in case of bladder, it is known as detrusor muscles. So remember the name, detrusor muscles. And finally, the outer layer. That means again, it's similar to your ureter. The outer layer is made up of fibrous connective tissue, and this is referred as adventitia. Fibrous connective tissue. So this is how the wall of your bladder is formed, and it helps in holding the urine for a temporary period of time. The bladder leads to the urethra, and the length of the urethra is different. The length of the urethra is different. In case of male, in males, the urethra is longer as it runs through the penis. And in case of females, the urethra is short. Now we will start with the structure of nephron. Okay, we will begin with the structure of the structure of nephron. Nephron. So the nephron is basically functional unit of kidney. It is also known as uniferous tubule, as it has lots of tubular structure. So I'm going to draw one by one. So the first part is a cup-like structure, and this structure is known as Bowmans capsule. It is known as Bowmans capsule. The second structure is a tubular, that is highly coiled tube. It is known as PCT. PCT stands for proximal convoluted tubule, as it is closest to the Bowman's capsule. That's why the term proximal. Proximal means close. The proximal convoluted tubule then leads to a loop which enters into the medullary region, and this loop has two limbs. It has two limbs. One which goes down and one which comes back up. So this white color structure over here, it is called as loop of Henley. Loop of Henley or Henley's loop. The one which goes down is known as descending limb. Descending limb of loop of Henley. And the one which moves up is known as ascending limb of Henley. Ascending limb of Henley. Further, this limb continues into again a coiled structure, coiled tubule, and this one is known as DCT. DCT stands for distal convoluted tubule. By the time the urine reaches to the distal convoluted tubule, it becomes concentrated, and it leads to once again a tubular structure which is common to other nephron as well. So that means this is one single tube to which many other nephrons might be attached, and this tube is known as collecting duct. So these are the part of the nephron. It starts with Bowman's capsule, then proximal convoluted tubule, which is a highly coiled structure. Then there is a loop formed here, white color structures. It has two limbs, that is descending limb of loop of Henley, this one, which goes deeper into the medullary region, and this lip, which is referred as ascending limb of loop of Henley. Further, it continues as distal convoluted tubule, and then it enters into a common duct called as collecting duct, which again runs into the medullary region. Give me 1 second and I will further show you that these parts which are above the dotted line are present in cortex region. So that means this much parts are present in cortex region, whereas below this line these parts are present in medullary region or medullary pyramid. Medullary region, medullary pyramid. We are going to see one by one how the nephrons are working in a kidney. Let us move on to the next section, that is types of nephron. So as I said, the kidney is divided into two portions. Suppose, let me write the title, types of nephron. Mainly the human kidney contains two types of nephron. Considering this, the partition of cortex and medulla, as I told you, this one is cortex and this one is medulla. So suppose that a nephron has its Bowman's capsule, its PCT and its DCT in the cortex region. So it appears to be the major section of a nephron such as PCT, Bowman's capsule, DCT, etc. lies in cortex region. Such type of nephrons are called as cortical nephrons. Cortical nephrons. So what are cortical nephrons? Cortical nephrons are the nephron that has most parts lying in cortex region. Okay, most part lie in cortex. Whereas if you see the other type of nephron whose Bowman capsule is also present in cortex region, whose PCT is also present in cortex region, but the loop of Henley is quite running deeper into the medulla and DCT, and once again the collecting duct is also running deeper into the medulla. So the major portion is present in the medullary region. Okay, so such nephrons are referred as medullary nephron or juxtamedullary nephron. You can call them as medullary nephron or juxtamedullary nephron. So why they are called as juxtamedullary nephron or why they are called as medullary nephron? Because most portion is in the medulla. Okay, so there are two types of nephron we have seen, that is cortical nephron and juxtamedullary nephron. Although we have 80% of cortical nephron and 20%, more or less 20% of medullary nephron. So the advantage of medullary nephron is that it is involved in most water reabsorption. Okay, so medullary nephron are actually made with the intention, or they are created with the intention to reabsorb most water and salt. Okay, which we will be looking further. Let's move on to the next topic, next section of this particular chapter. So once again we are going to see the nephron structure, but this time with the blood capillaries. That is, you must have remembered the hilum region, that is the portion through which blood enters, that is renal artery enters. The renal artery enters and then it gets divided into arterioles. So there is one arteriole which enters into the Bowman's capsule. It enters into the Bowman's capsule and it has a thick diameter. The lumen, the diameter of the lumen is little larger. It enters into the Bowman's capsule, the dent or the notch like structure, and then it exit as a narrower tube. So this portion, this one, which enters into the Bowman's capsule, it is referred as afferent. A for afferent, A for arrival. So you can remember like this, that it arrives into the Bowman's capsule. So afferent arteriole. It is called as afferent arteriole. Then it enters into that notch-like structure, which will be referred as glomerular capillary. It is referred as glomerular capillary. These capillary have tremendous pressure which causes the filtration and the blood gets filtered and then it enters into the capsular space. We will discuss this later. Now it exits as this tube which is little narrower, and this one is referred as it exists, E for exits, it is referred as efferent You can remember like this: efferent arteriole. So the blood enters via afferent arteriole and it exits as efferent arteriole with a narrow lumen. So the lumen is narrower in this case. Just give me 1 second. Narrow lumen, and in this case it's wider lumen. Now this blood capillary then moves around PCT, DCT, moves around DCT, and somehow it comes back to the loop of Henley, and then slowly this gets converted into venular structures, that is veins, and enters into main renal vein. So we're not concerned right now that thing, but just we have to remember that it forms capillary around the PCT DCT, as well as it forms the capillaries around the loop of Henley, something like this. So what are these red color structures referred as? The capillaries which surrounds loop of Henley are referred as vasa recta. This is very much important, as vasa recta is one of the important element or component for water and salt reabsorption. The water and salt which are actually taken from the loop of Henley enters into this vasa recta. So that means in water conservation, vasa recta is also playing an important role. What are these capillaries called which are present around the PCT, that is coiled tubules, PCT and DCT? Both these PCT and DCT capillaries, I mean the capillaries which are surrounding the PCT and DCT, are referred as peritubular capillaries. Peritubular capillaries. They are referred as peritubular capillaries. Okay, they both are referred as peritubular capillaries. So whatever the reabsorption is going to happen in PCT and DCT, that will be dumped or that will be reabsorbed by these capillaries. So the whatever thing is lost will be retained again into these capillaries. I hope you understood this one. Let's move on to the next section. Now we are going to see one by one what is Bowman's capsule, what is the importance of this Bowman's capsule. As I've already told you, the blood enters with a broader lumen. So the pipe is going to enter. It will be forming a numerous tuft and it will be exiting as small capillary. And as soon as it enters into the Bowman's capsule, it gets divided into numerous network-like structures, and this is referred as glomerulus. This is referred as glomerulus. Now, this is glomerulus, and this is Bowman's capsule. Bowman's capsule. Together, glomerulus and Bowman's capsule. Okay, you need to be very clear about these all terms. Together, glomerulus and Bowman's capsule is referred as Malpighian body. Now, listen carefully. Previously, we had discussed Malpighian tubules. Malpighian tubules are the part of your cockroach, but these structures, that is glomerulus and Bowman's capsule, form Malpighian body, and this is the part of nephron. So there is a big difference between Malpighian body and Malpighian tubule. Do not get confused with the cockroach Malpighian tubules or insects Malpighian tubules. That is something else. This is something else. Hope this is clear. Once again, I'm repeating. Just be careful about this naming. Okay, the one which enters, the one through which the blood enters is referred as afferent arteriole, and it divides into glomerular capillaries, or glomerulus, and then it exits as efferent arteriole. Hope it is clear. So I hope there is no problem in understanding this. This difference in lumen is the main reason for the pressure required for filtration. Pressure. So narrower lumen and wider lumen difference creates the pressure because of which the filtration will occur. So as you can see, it's a normal physics phenomena applied. The filtration occurs. The filtrate enters in capsular space. Now we will see how it filters. That is, if you see at the Bowman's capsule, what you will find is there are two layers. The inner one is formed of a special cells. They look something like this. So I'm not going to extend this. These are actually a type of modified squamous epithelial cells. As you know, squamous epithelium cells are required at the site where filtration is supposed to happen or exchange is supposed to happen, and here the filtration is happening. So this particular cells or this particular layer is called as visceral layer. Visceral layer of Bowman's capsule, and it is composed of special cells called as podocyte. Podocytes. Podocytes. And the podocytes are the major cells which are responsible to cause filtration, because podocytes bear slit pores, whereas at this layer, that is the one which is surrounding the outer layer, these are simple squamous, the typical one which you have already studied in the chapter structural organization. And this layer is called as parietal layer, which is made up of simple squamous. So both are squamous, but the inner one is specially modified because it should be functioning for the ultrafiltration process. So it is suitable for ultrafiltration process. Simple squamous, and podocytes bear slit pores that aids or that helps in ultrafiltration, which we will be discussing in a moment. So I hope this is clear what is present inside the Bowman's capsule and how it is made. This space is called as capsular space. Capsular space. Okay, fine. Now when the capillaries enters into the capsule, even capillary bears squamous epithelium, which are specifically referred as endothelial lining. This is called as endothelial lining. So I'm not making all the capillaries, but just for understanding purpose, as you can see, squamous epithelium. And between these layers, that is between the endothelium, now this one is referred as endothelium. So blood is going to cross, the plasma is going to cross the endothelium first, then there is one more layer present between the podocyte layer and endothelium layer, and this layer is called as basement membrane. This layer is called as basement membrane, which is completely interfering between these two layers. This layer is of basement membrane. So endothelium, basement membrane, and visceral layer. I'm repeating once again that these three layers, that is endothelium of blood capillary, basement membrane, and visceral layer of Bowmans altogether form filtration membrane. Hope this is clear. Now we will move on to the next session. Okay, so as I was talking about the podocyte, let us zoom into the visceral layer, how a podocyte looks like. So just for reference purpose, I'm going to make like this. It looks something like this. So it has very narrow enfoldings. Do not get confused with brush bordered epithelium. They are not, because brush bordered is only present in your cuboidal or columnar epithelium. And as I already mentioned, these cells are squamous, modified squamous epithelium. These are modified squamous epithelium. So these structures, each cell has this flat protrusion which are called as pedicels. Pedicels. And the space between these pedicels, here I will be drawing that with yellow color. The space, this space is referred as, through which the filtration will be occurring. These spaces are called as slit pores. This is the thing which is going to help in the filtration. These are referred as slit pores. They are very fine. And this one is the nucleus. This is nucleus. So this is a typical structure of your podocyte. What are podocytes? Podocytes are modified squamous epithelium present in the visceral layer of your Bowman's capsule, and they help in ultrafiltration. Modified for ultrafiltration. Modified for ultrafiltration. Hope this is clear. I'm just covering important differences or important minute details. So please be careful about each and every term. Let's quickly zoom into the PCT. So we have seen how Bowman's capsule is arranged. Just give me 1 second. So this is PCT. And we are going to zoom this PCT portion. We're going to zoom this PCT portion. So what do we see in PCT portion? That it is lined by cuboidal epithelium. It is lined by cuboidal epithelium, but not a simple cuboidal epithelium, a special one that contains microvilli. So what does a microvilli suggest? That means these are going to perform absorption. These are going to perform absorption. So this is the lining of the PCT, and it is lined by brush bordered cuboidal epithelium. Brush bordered cuboidal epithelium. What is special thing about these cells? Firstly, they're cuboidal, as I have already mentioned, but second thing is that they contain numerous mitochondria. And as the number of mitochondria are high over here, this suggests that they perform active reabsorption, because mitochondria produces lots of ATP, and ATP is required for active reabsorption. So active reabsorption is seen over here, and absorption is somewhere around 70 to 80% will be absorbed here. 70 to 80% of nutrients is absorbed here. In PCT. Okay, 70 to 80% is absorbed over here. The next part is loop of Henley. Already I have drawn the diagram and you can go back and check once again, but I'm going to just zoom on this part. So consider this is the loop of Henley, and there is no error in the drawing, but as you can see, this portion, let me draw it with some other color, this portion, and observe this portion. There is difference between the lumen. So let us zoom first this one. And as you can see that this is thin as compared to the other one, and it is lined by squamous epithelium. This is lined by squamous epithelium, and this one is referred as descending limb. Okay, this one is referred as descending limb. So descending limb of loop of Henley has squamous epithelium which favors, okay, which favors water exit. So water will be getting out of this tube into the nearby capillaries. So permeable to water, but almost impermeable to salts. Okay, now let us zoom this portion. So once again, when we closely observe over here, this is little wider and it has cuboidal, not squamous, it has cuboidal epithelium. It has cuboidal epithelium, and this cuboidal epithelium is responsible for active sodium reabsorption. That means the sodium will be dumped into the interstitium of medulla. Sodium, not water, but sodium. So let us see what I said. Basically, the thin, that is descending limb, is responsible for permitting the water into the interstitium, whereas the descending limb, okay, this one is this one is ascending limb, ascending limb of Henley's loop. It is permeable to, okay, it is permeable to electrolytes, but not to the water. It does not allow water to exit, but only to electrolytes. So this was about loop of Henley. Hope you understood this portion. of the topic. Now the DCT. As compared to your PCT, DCT does not show presence of microvilli, and it is also less supplied with mitochondria. So suppose this is DCT, they have cuboidal cells. DCT has cells with less mitochondria, and they are involved in reabsorption of water and salts under influence of hormone. That means in the presence of hormone the absorption will occur. So suppose this is the peritubular capillary present around the DCT. So if the water absorption has to occur, so water will be entering into capillary, and even the sodium or other electrolytes will be entering into the peritubular capillary, but that will only happen facultatively, that is conditional, under the influence of hormones such as maybe ADH or aldosterone. So hope this is clear. This is the thing that will be happening in DCT. This is the thing which will be happening in DCT. Okay, let's move. So in the today's video, what we have learned is about structure of kidney. We have seen structure of nephron and the individual parts of the nephron, and again its types, that is cortical nephron and medullary nephron. In the next video we will be looking at the process of urine formation. So please go through it, revise it, and learn it and practice the question. Till then, thank you for attending the session. See you. Bye.